Hello, everyone. Our lab is interested in programmed cell death, which is a strictly scheduled, genetically encoded, and tightly regulated cell fate. It is a widely observed and evolutionary conserved trait seen during normal development and throughout the life of the organism. Programmed cell death has many functions in animal development. For instance, it is important for refining or eliminating structures as well as quality control like eliminating abnormal, misplaced, or dangerous cells. Programmed cell death has been extensively studied, especially in simple cells with simple structures, but this is not true for morphologically complex cells that are commonly found throughout an organism. How do these complex cells die, and how are these different compartments killed and removed? One of my favorite examples is found in the nervous system, which has a neuronal removal and refining system called pruning. This involves selective and targeted removal of axons and dendrites. Unfortunately, the molecular mechanism of programmed cell death in morphologically complex cells and pruning is not well known. And while in vivo studies show critical insight, they pose technical challenges. To address our questions, we are using the model organism C. elegans, which has numerous advantages. Importantly, we can perform in vivo studies. We are studying a phenomenon that takes place in C. elegans called compartmentalized cell elimination, or CCE, which is a novel program of cell death we discovered. In CCE, different parts of the cell are treated differently and are eliminated differently. The tail-spike cell is a morphologically complex epithelial cell that is located in the tail of the worm, and it is involved primarily in tail formation in the embryo. And once it reaches a certain stage, the tail-spike cell undergoes CCE. In the first step, the cell has started to die, and you can see the soma has rounded like an apoptotic cell, and the process has severed. Here, the compartments are a little bit more evident. Next, the proximal process is gone, and you can see the soma has shrunk as well as the distal process has retracted into itself and are left with two small pieces that will be eliminated separately. Importantly, by the time the animal hatches, the entire cell is completely gone. This fascinating death program is also observed in a set of sex-specific sensory neurons called CEM neurons that die in the hermaphrodite but live in the male. It is amazing that these two cells on polar opposites of the worm in different environments and locations are dying the same way. What this shows is the idea that CCE is potentially universal and may be a broad phenomenon observed throughout an organism. Something interesting and important that we observed in our CCE system is the similarities to pruning. We see fragmentation in the proximal process and also something like pruning in the retraction of the distal process. And so we see two pruning-like events in the same cell process, which allows us to potentially learn more about the molecular mechanisms of pruning, all by studying one cell. This leads us to my study. We found that stability genes LNP1 lunapark and ATLN1 atlastin are required for CCE. Lunapark and atlastin have been shown to stabilize three-way junctions of the smooth ER, and when observing mutants for these genes, we were able to see a bit of tail spike cell remaining. We then wanted to know if LNP1 functions in the tail spike cell. To address this, we performed a cell-specific rescue experiment, and interestingly, we found that while control animals showed the expected tail spike cell persistence, the transgenic animals expressing wild-type LNP1 showed significantly less of a defect, showing a rescue. This data suggests that LNP1 functions autonomously in the tail spike cell. Next, we found that LNP1 is expressed in the tail spike cell. You can see tail spike cell in green, and magenta is the LNP1 promoter. We repeated this experiment but tagged ATLN1 promoter instead and found similar results. From the literature, we learned that atlastin has a binding partner, which is an ATPase called spastin. Spastin is a microtubule severing protein, and we then observed a SPAS1 mutant, which gave a very similar CCE defect phenotype to LNP1 and ATLN1. Next, we found that SPAS1 is also expressed in a tail spike cell, just like LNP1 and ATLN1. After this, we created and observed ER, SPAS1, and EBP2 reporters. When comparing the ER and SPAS1 protein localization, we can see similar enrichments in the proximal process, while the microtubule plus N binding protein EBP2 seems to be enriched in the distal. Some of the upcoming experiments we have involves characterization of our markers of wild-type and mutants via microscopy, as well as cell-specific rescue of elastin and spastin. This data leads us to our model that we are proposing. This involves a stable ER, which then allows for precise positioning of spastin that leads to the cleaving of microtubules by spastin, and allowing for the organized assembly of microtubules leading to CCE. But when ER stability genes or spastin is mutated, microtubules are hyperstabilized and CCE cannot take place normally, leaving us with remaining tail spike cell. Thank you very much.